Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is David Chen, and I'm a postdoc at New York University, and will be moderating today with Ceri Weber, a postdoc at the University of California, San Diego. We are excited to highlight the work of our outstanding postdoctoral members. Today, Ashley Libby from the Francis Crick Institute and Silas Boye Nissen from Stanford University will share their research. Each speaker will give a 20-minute talk, followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. Our first speaker today, Dr. Ashley Libby, is a postdoctoral researcher in the Briscoe Lab at the Francis Crick Institute. She earned her Bachelor's of Arts in Molecular and Cellular Biology from Vanderbilt University, with minors in mathematics, French, and chemistry. She completed her PhD in developmental and stem cell biology at the University of California, San Francisco, as part of the Gladstone Institutes, working under the mentorships of Drs. Bruce Coughlin and Todd McDavitt. Her doctoral research focused on gastrulation patterning using induced pluripotent stem cell models, developing CRISPR-based assays, machine learning classifiers, and 3D organoid systems to study self-directed multicellular organization and morphogenesis. Currently, Dr. Libby investigates the genetic mechanisms underlying tissue production and stem cell fate determination in the caudal epiblast during neural tube development. Her work combined in vivo CRISPR technologies and live imaging to uncover how vertebrate neuron tubes arise from the stem cell populations. Dr. Libby received several prestigious awards, such as EMBO Long-Term Postdoctoral Fellowship and the NIH NRSA F31 Predoctoral Fellowship. Dr. Libby is also a dedicated mentor, guest lecturer, and advocate for scientific outreach. It's a great pleasure to welcome Dr. Libby to today's seminar, where she will share her research in neural tube development.
Thank you very much, David, and thank you everyone for coming and the SDB for having me. So today I'm going to be talking about crispy chickens, but really I'm going to be talking about CRISPR in vivo screens to look at regulation of neural tube fate acquisition, and specifically a gene that we found of interest. And so one of the first things I want to simply refresh, I know this is developmental biology, but I find it absolutely fascinating that we can build complex tissues starting from a limited number of cells. So what you're watching on the right is a video of chicken development, where you have the head fold forming and then the spinal cord extending down slowly from there, being built over time. And we know that this embryonic development starts with a limited number of cells, but then moves into these complex tissues that build and grow, where you eventually have a full organism. And what the genetic and physical mechanisms that allow for this complex tissue generation is what I have always been fascinated in. And I hope to convince you that by the end of this talk, the developing spinal cord is a really great system to kind of look at this. And this is in part because our entire trunk development has a series of fate decisioning and patterns that have to be made. So first, cells in the back of the embryo, here in what we call the caudal epiblast, have the choice of two fates. They can decide to stay within this caudal epiblast, which is kind of like a plate. This then rolls into a cup and then closes into a tube. And this tube forms the neural tube and eventual neural crest. It will go on to form the entire central nervous system. Now, the cells in this caudal epiblast also have a second decision they could make. Instead of staying within the plate, rolling to the cup and tube, they can instead dive through the primitive streak and migrate around to the sides of this cup and tube to form the mesodermal tissues. So the paraxial mesoderm, which go on to form the somites and vertebra, the intermediate and lateral plate mesoderm. And this decision, we know, is incredibly important, but happens in a very dynamic environment. So not only do these cells experience multiple signaling molecules while they're in this plate making the decision, but as they move out of the plate into the cup and the tube, they experience a change in morphogen exposure. So the plate has high levels of FGF and Wnt in the back of the embryo, and you move into a retinoic acid high signaling center as you roll into that cup and close into into the tube. And at the same time, there are the emergence of signaling centers in the form of sonic hedgehog from the floor plate and notochord, and BMP from the extraembryonic ectoderm and eventual roof plate. And these change and physically move over time as the embryo is forming. In addition to this, we know that there are specific gene expression programs that cells have to decide to turn on and off to become certain lineages over another. And so how a cell can interpret all of this information, rapidly change its gene expression, and then physically move into place is a really fascinating question. And so what we set out to ask at the beginning of my postdoc project is, how does early reception of these signals affect our later fate decisions? And then what are the gene regulatory mechanisms that control this rapid and efficient lineage acquisition? And so one of the ways that we originally decided to address this is simply to observe what is going on in the back of the embryo. And for this, I worked collectively with several postdocs in the lab to characterize the progenitor to neural fate transition. So specifically to look at what happens going into the neural tube. And we generated a single cell data set across time of multiple ages of chicken embryos, and then ask, okay, what cell types do we see? What gene expression patterns do we see? And so this, to orient you to begin with, this is a UMAP of single cell RNA data, sequencing data. Every dot represents the genome of, or the gene expression profile of an individual cell, and similarly colored dots mean that they have similar gene expression profiles. So now what we can do is we can take this data set and we can orient ourselves in terms of defined gene expression that we know in space. So for example, we know in the caudal epiblast, you have high expression of a gene called TBXT, also known as brachyury. And then as you move into the neural tube, you're going to see concentrated expression of a gene called PAX7. And so we can map these two genes onto our data set and make a general idea or hypothesis that one side of the data set is going to be mesoderm and the other side is going to be moving towards neural tube, and presumptively in between, where you lose that brachyury and transition to neural tube, is our caudal epiblast. And so what we can do is we can then ask, okay, once we've set up ourselves within the data set, now what happens with genes that we don't know a lot about? So for example, here, what I've just done is I've reoriented ourselves, and this is a heat map plotting our mesoderm on the right and our neural fates on the left, and then the gene expression where yellow is very high gene expression. And what we can see is, once again, we can look at our TBXT or brachyury, and you can see that it's expressed in between definitive mesoderm and the neural tissue. We can go back into the embryo and stain using HCR for brachyury transcripts and show that they are back here in this caudal epiblast region. And then this is a maximal projection, so you actually see the notochord underneath the neural tube here. Now we can then ask, okay, what happens to genes that we don't know a lot about? So for example, this is CMTM8, which is involved in the c-Met pathway, and it is expressed a little farther towards the neural lineage in our data set as compared to brachyury. And if you go back to the embryo and you look at these transcripts via HCR, you instead of just seeing concentration around the back of the caudal epiblast, you see this extension of gene expression into this cup region of the primitive or the pre-neural tube. And what we can do is we can then take this information to generate new molecular maps of the caudal epiblast and the transition into neural tube with novel genes or genes that we had not necessarily been focused on before. And so from here, we can ask, okay, what are the genes that mark this transition of moving from this plate to cup to tube? And are they, one, do we know that they should be important in this process? Are there novel genes that we want to look at? And how are we going to do this? And so we decided to move forward by selecting a small subset of 25 genes that all mark this boundary moving from this plate to cup to tube. And we decided to do an in vivo CRISPR screen. And what we can do here is pool our knockouts into one embryo so that you can knock out 25 things at once, and then use single cell sequencing to pull out what single cell has gotten what knockout at what time. And so the way this works is we have a 2-cassette Cas9, CRISPR-Cas9 system where you have Cas9 attached to a citrulline protein, and then you have your guide RNA with a chick-specific promoter. This can be electroporated specifically into the back of the embryo. And what we do is we wait for 24 hours. We can then FACS sort for everything that's gotten the Cas9 protein and use single-cell RNA sequencing to pull out what knockout is in what cell. And we can do this because we've added a 10x capture sequence into this guide RNA, which is extremely useful because then you can figure out what knockout ends up where. Now, questions that we want to ask is twofold. One, if you knock out a gene, what is its enrichment in a certain cell type or depletion in a certain cell type compared to scramble? Meaning you cannot become a certain cell type if you lose a certain gene. And then the other question is, if you knock out a gene, what other genes are misregulated? So it's kind of twofold. And the first one I'm going to talk about is enrichment and depletion. So when you look at enrichment and depletion, what you can generate are heat maps that look like this. So this is a heat map where all of the columns represent different cell types in our data set. So I've labeled them with their broad categories of caudal epiblast, surface ectoderm, neural tube, primitive streak, and then another neural tube category. And then every row here represents a different knockout. So for example, what you can tell is there are two main trends. Whether you are enriched or depleted in the caudal epiblast, or whether you are depleted from neural tube and primitive streak, so more definitive things. So for example, if you look at brachyury, TBXT, which is our main kind of internal control, if you knock out brachyury, you're depleted from the caudal epiblast, which makes sense. We know that brachyury is highly expressed there, so if you knock it out, the cells leave the caudal epiblast. And then you find that they're also enriched in neural tube, which also makes sense because as of right now, we don't presume that brachyury is necessary in neural tube formation. But what we can do here is pair this information with what is also happening at a gene regulatory level to ask what is interesting, what is going on. And the way that we decided to first tackle this is to look for overarching themes. And we did this by taking gene ontology terms of the genes that have been misregulated for each of these knockouts. So what you're looking at here is actually similarity of gene ontologies between each knockout. And so something that I'm going to draw your attention to is that there are 4 genes that share very similar gene ontology terms or pathways that are enriched or depleted when you knock out this gene right here in the center. And these are MLT3, CMTM8, GREB1, and SPRI2. So now we know that SPRI2 is associated with FGF signaling, GREB1 is associated with retinoic acid and other signaling. CMTM8, I've already told you, is a part of the CMET pathway, which is also another form of signaling. But MLT3 stood out to us because not much is known about it in terms of signaling environments, but also in terms of development of the neural tube. So we decided to look into this a bit further. So if we go back to our embryo and we simply just stain again for the transcripts of MLT3, you can see it's highly expressed in this plate region of the caudal epiblast. And when we look at our data set and ask exactly what genes are enriched or depleted, misregulated from compared to just a scramble when you knock out MLT3, we see that there is a depletion of genes associated with retinoic acid, such as RORA1 or ROR2. We see that there's misregulation of the Wnt pathway, specifically the PCP pathway with Wnt5A, Wnt5B. But then we also see that there's an upregulation of FGF receptors and BMP signaling molecules. And so what it seems is that this particular gene and its misregulation sits in between multiple pathways that we know are involved in the caudal epiblast and then transition out of the caudal epiblast. So if you remember, in the back of the embryo, you have high FGF and Wnt, and then you move into a high retinoic acid. And so we decided to look at this a little more closely. But first, what is MLT3? So MLT3 is part of the superelongation complex, which is an epigenetic complex that helps to unpause and activate transcription initiation. So at certain times in development, polymerase II is paused at the start of different genes as if it's waiting to be activated. And what activates it is this superelongation complex, which comes in and phosphorylates polymerase II and allows for transcription to start. Now, MLT3 is part of this complex, and it's the epigenetic reader that helps to direct the complex to specific histone marks, so specifically histone chronotinylation and acetylation. Now, we know from previous studies that this particular gene, it's actually quite important for hematopoiesis, both in just hematopoiesis stem cell self-renewal and engraftment, but also in the generation of different types of hematopoietic cells. And then there's a small amount of literature looking at it in cranial neural differentiations in vitro. And so we thought that this was an interesting thing to follow up on. And the first thing that we decided to look at is what happens when you just knock out MLT3 only. So everything I've been talking about before is in a pooled screen manner where we're looking at 25 things at once. So if we just go back to the embryo and only knock out MLT3, once again in the caudal epiblast, what happens? And so here what we're going to be doing is we take our Cas9 and our MLT3 guide, and then we also have an internal control to make sure we've hit the right spot, which just has M-scarlet fluorescence. So one of the first things that we noticed, if you compare a scramble or a control to an MLT3 knockout, what you're looking at here is the neural tube labeled by SOX2 HCR, and then the tail bud labeled by brachyury. We can see that there is a kink in the spine, or these neural tubes are not necessarily straight. And we can actually quantify this by looking at the ratio of perimeter to area of the neural tube, or what takes up the neural tube. And we can see that with MLT3 knockout, you no longer have a very straight neural tube, but in fact, it's quite wavy. So why might this be occurring? Well, if we look at the neural tube in these embryos and compare our scramble on the top here with our MLT3 knockout, what we noticed is that within the scramble, you see MLT or scramble and Cas9 cells labeled in blue can make it into the neural tube just as much as our control cells labeled in red. However, if you look at the MLT3 knockout, there's a large depletion of the blue cells, or these knockout cells, that make it into the neural tube. And we can see this in whole mount, but in order to quantify it a bit better, we also looked via flow. And once again, we see that there's a loss of cells moving into the neural tube as marked by SOX2 neural progenitors after dissection of these embryos. And so where are these cells going? Well, you get a little hint here in this bottom line. You see the stream of blue cells outside of the neural tube domain alongside the somites. And so in fact, these cells, instead of moving into the neural tube, end up in the lateral plate mesoderm, which, if you remember, is the farthest over mesoderm from the neural tube. And we can go back and section the embryo and show that these blue cells are once here all the way in the lateral plate mesoderm. We have some in the intermediate mesoderm, and we have a rare few that make it into the somites and neural tube. So something about losing MLT3 prevents you from moving into a neural tube lineage. So that's cool, but why? Why would losing an epigenetic regulator that starts transcription prevent you from becoming neural rather than and pushing you towards a mesodermal fate? And so for here, we went back to the literature to one of the papers that had looked at this in the brain, and specifically in human stem cell differentiations in a dish. And they made the claim that MLT3 actually can bind as a cofactor to retinoic acid receptors. And in the presence of retinoic acid, direct the superelongation complex to retinoic acid responsive genes, which in terms of the caudal epiblast are the neural responsive genes, or the neurogenic responsive genes. So here is a great example of collaboration in science, because I wrote to the last author of this paper that was published over 10 years ago, and he dug through his -80 freezer and immediately sent us a series of plasmids to test this hypothesis. So what he did in his paper, and what we repeated in vivo in ours, is you have a series of knockout plasmids where you have a full overexpression cassette of your retinoic acid receptor alpha attached to an M-scarlet. Then you have an overexpression of just the retinoic acid receptor without the binding domain of this MLT3, and then an overexpression of just the binding domain. And what this would allow is to test two different ways that MLT3 would be prevented from binding to retinoic acid receptors, and therefore prevent this recruitment of these superelongation complex to these genes. So what we would predict is that the overexpression of these would competitively inhibit our lineage differentiation towards the neural tube. So now we can repeat the same experiment So I'm repeating this experiment. I'm once again showing you flow, or an average of our flow plots, where you have a scramble control, and this is the amount of cells that have made it into the neural tube. And if you knock out MLT3 here in pink, you see a reduction in the cells that are able to then move into the neural tube. Now, if we overexpress our regular retinoic acid receptor, we see no difference in cells making it to the neural tube as compared to scramble. But if we overexpress either of our competitive inhibitors, we once again see this drop. And this is really interesting because what we think is happening here is that the embryo is using this spatially restricted transcriptional regulation to drive this rapid acquisition of fates. So in the back of the embryo, where you need to be very sensitive to the appearance of retinoic acid, you have a high amount of MLT3 that then the moment retinoic acid is sensed, it can switch on your neurogenic genes and force that cell to move into the neural tube or to choose the neural lineage so that you can very quickly move into that cup and close into that tube, forming the correct cell types in the correct timing. So, in summary, what I've talked about today is how we've taken a single-cell RNA sequencing data set to refine our knowledge of transcriptional domains or the molecular geography of the embryo. We've then used this to help develop an in vivo CRISPR screen that pools a bunch of knockouts together into one embryo and looks at aspects of gene regulation within trunk fate decisions. And then through the screen, we've identified MLT3 as a regulator in this movement into the neural tube, in the decision to become neural cells. And this is importantly done with through an interaction with retinoic acid receptor alpha. And so kind of to leave you on this, what I like to think about in the future is how does this initiation of transcription and rapid transcription, how can this information help us understand control over rapid fate decisions in other tissues in development? And is it just in the neural tube, or do we see it going the other way as well? And do we see it in other tissues? And with that, I'm going to finish here. I'd like to acknowledge my lab. I work in the Briscoe lab at the Francis Crick Institute. Specifically, I'd like to acknowledge Tiago and Arthur, my co-authors on this paper, Despina and Julia, the other chicks in the lab, and our fearless leader, James Briscoe, my funding, and I'm happy to take questions.
Thank you, Ashley, for this beautiful talk. While we are waiting for the questions to be typed in, I will first ask a question. So your study identified as MLLT3 as a key regulator of neural fate acquisition. Do you think this role of MLLT3 is conserved across species? And how about other parts of the tissues?
So we know that the only even reason we looked at MLT3, or not the only reason, but one of the large reasons we looked at MLT3 is because we saw it in our expression, but then it had also been identified in both mouse and human tissues as well. So we know that in the brain, at least, MLT3 is quite active. We know that in human pluripotent stem cells, as they move into a caudal epiblast phenotype, you have MLT3 expression. So we know it's conserved across species. I have preliminary data looking at it across different tissues. So we know that it's expressed in parts of the brain. We know that it's expressed in the eye and forming eye cup. And then, of course, we know that it's expressed in early blood islands and hematopoiesis.
Okay, thank you. I got another question from Bharat Esturi. Have you created any GRM model for this result?
So no, we haven't yet created any gene regulatory network models. And this is in part because MLT3 hits many things, or at least the super elongation complex hits many things. And it's not necessarily often gene regulatory network models are established between transcription factors that can turn on and off other genes. And so MLT3 seems to be facilitating the already established retinoic acid based transcription gene regulatory network. So we haven't yet elaborated on that, but stay tuned.
Yeah.
The Briscoe
Okay.
lab will move into it.
Another question from the panelist, Martha asks, do MLLT3 mouse mutants display neural tube patterning defects?
In fact, they do. So no one has ever looked to see if you have patterning in different neural tube cell types, but they do present scoliosis, kink spines. They have fusions between their different vertebrae. So there's clearly something wrong in just the generation of the correct proportion of tissues between mesoderm and neural.
So another question from the panelist, Billy. She said, beautiful talk. Actually, it's very interesting that the cells that lack MLLT3 move to the lateral plate mesoderm. Do they later differentiate into muscle?
So again, I don't know this particular answer, in part because we don't let the chickens grow up that long. So I have yet to follow up on whether they move into a complete incorporated muscle or if they are maybe stalled, and that the lateral plate mesoderm is almost like a storage area for them, and then they'll eventually be discarded because they are either unviable to the embryo, etc. So I don't know the direct answer to that yet. Yeah.
Right. She commented like it would be interesting to see what happens to them. Yeah,
Yeah.
agree.
Stay tuned.
So I have another question while we're waiting for other people to type in questions. So do you think, so retinol acid signaling, do you think that's the main source to activate this MLLT3 pathway, or is this the right signal? So like the upstream is retinol acid. If you knock it down, do you phenocopy the MLLT3 knockout?
Right. So we tried to do this. We had an internal retinoic acid receptor alpha knockout control, which did not phenocopy this. In fact, the embryos were perfectly fine if you knock out this retinoic acid receptor alpha. Unfortunately, there are a lot of different, and we know this from mouse knockouts and in embryos, that there are a lot of ways that the retinoic acid receptor or the retinoic acid pathway is redundant. So it's hard to use a receptor to phenocopy this. What we do know in terms of other signaling pathways is that this complex, not necessarily MLT3, but the complex itself interfaces with Wnt signaling in hematopoiesis, and it interfaces with FGF signaling in a lot of different cancers. And so we know it sits at the center of multiple signaling complexes or signaling regimes, one could say, and that perhaps retinoic acid is the one employed here to distinguish between neural and mesoderm. And we haven't even looked really in depth at the mesoderm portion of this, so it could be that Wnt is driving it the other way and that retinoic acid is driving it towards neural.
Okay. So there's another question from the panelist. Ceri asked, as a tail person, I'm fascinated by the trunk-tail transition. What do you think happens if you were to sample the chicks at the end of axial elongation?
So I do know this in general. So MLT3 is expressed all the way into the tail bud. It's really strongly expressed in the tail bud. And so you still get it all the way up until the end of your axial elongation. And so that particular, I guess, the decision using MLT3 in this elongation complex is not specific to early primary neurulation. It also occurs in secondary neurulation. And similar to brachyuria expression, MLT3 fades out over time as you come to the end of the use of the tail bud in your axial elongation.
All right. And there's no more question. Let's thank Ashley for this beautiful talk. And I'll hand it over to Ceri, our second moderator. Thank you.
Thank you.
Hi, everybody. I am happy to introduce Dr. Silas Boye Nissen from Stanford University. Silas has been training in physics and biology since his undergraduate training at the University of Copenhagen, culminating in his PhD in biophysics from the Niels Bohr Institute in 2020. He has received considerable funding and fellowship prizes to support his research on biophysics and developmental and quantitative biology. And while reading his CV, I was struck by his research and training experience around the world and clear commitment to his academic community and field. Not only has Silas spent time as a visiting scholar at universities around the world, but he sat on multiple committees, given numerous international conference talks, communicated science to his community, and been a contributing author on many publications. Needless to say, I am excited to hear his seminar.
Thank you for that very kind introduction, and thank you for having me speak here today. Thank you to the organizers for organizing this fantastic series throughout the year. So look at this fly wing and notice how one hair comes out from each cell and points almost perfectly towards the tip of the wing. So the question is, how does nature do this? How does each individual cell break the symmetry and make one hair point towards the tip of the wing? How does the cell know what's left and what's right, or what's proximal versus distal? So you can think about a party and with a lot of Pepsi cans when the party is over, and you could either trash these or you can start orienting them upside down, and you will then have giving the Pepsi cans apical basal polarity. You can also order these to a second degree, left-right asymmetry or proximal distal, here with the tip of each Pepsi can representing one cell, and then the tip from the Pepsi can could, in this case, be representing the hair, that for the wing tissue, it comes out from the distal side of the cell and it points towards the tip of the wing. Okay, so here, this was after a party, but the question is, how did the cell or the biology do this same effect? And why do they at all care about doing this? So a planar cell polarity has known implications in neurotube defects, neurotube development. We heard a little bit about that in the previous talk. Heart development, where you have conotruncal heart defects when PCP is mutated. Cilia dyskinesia, also hair directions. Situs inversus, where the organs are not on the heart is not on the left-hand side, but on the right-hand side. And then deafness, where also PCP has known effect. And then PCP has known implications also for wound healing and cancer. So you cannot build a person without PCP. And not only that, the set of core proteins found in this signaling pathway are conserved from flies to mammals. Here from wild type tissue at the wing, you see beautiful hair directions, whereas in PCP mutants, pretty much when you remove any core PCP proteins, we find swirls in the hair pattern. And then here the example from mice, we see the inner hair cells in the inner ear, they are oriented almost perfectly up in wild type, whereas in PCP mutants, here CRAS mutant, one of the point mutations in one of the core proteins, mess up the orientations of the inner hair, and this mouse would lack here and also balance. So PCP is central to development.
Silas, I'm so sorry to interrupt you. Could you close the alert at the top of your screen?
Sure.
Thank you. Sorry.
It's this one, right?
Yes.
I can try at least. Is it gone?
No, it seems stuck. Don't worry about it. Oh, there it goes. Okay.
Okay, cool. Okay. So imagine your favorite tissue. You could pretty much, this applies to many tissues, not all, but here specifically the fly wing, but you could think about other tissues. At the end stage, we have hairs that comes out and they point in a specific direction. Initially, there are no hairs. And if you now look at two cells from the side view, we see them here on the left-hand side. What you will realize during this talk is that we have a set of proteins that form clusters that span the intercellular junction. Here, what ends up with like a set of proteins on the proximal side inside a cluster. And this is where we find two proteins, Wang and Prickle. You don't have to remember all the proteins. I will concentrate on the membrane proteins. So that would be Wang. And then one cytosolic protein, Prickle, that ends up in the end stage on the proximal side of the cell. So similarly over here, that would correspond to over here in the first cell. And then on the other side, we have two other proteins in this scenario here. We have a membrane protein Frizzle, and another cytosolic protein is disheveled that in the end stage accumulate on the distal side of the cells. And then to bridge these two subcomplexes, the distal subcomplex with the proximal subcomplex, we have flamingo that connects these two groups of proteins to each other across the cell junctions. So this is what builds a proximal distal axis that's perpendicular to apical basal and eventually orients the hair coming out from the distal side, pointing distally, and here reflecting back to the PepsiCan example from earlier on. So to give you real images and show that the cartoons that were before is actually the case, you can look at these 8 cells here from a fly wing tissue fixed sample, where we tag one protein weighing in YFP here on the right-hand side. And in the neighboring cells, we tag the same protein also weighing in CFP. And this allows us to make mosaics in the tissue, where we can now study the junction where these two tags, they meet each other, because now we can uniquely identify that the green that's coming down this junction is from this cell specifically, and the purple that comes down the same junction is from the cell on the other hand side specifically. Okay, so what we see here is that the weighing accumulate into puncta, or into clusters as introduced earlier, with the highest degree of accumulation on the proximal hand side. Here you see a line scans going down this junction here, so you see the 3 clusters with high accumulation of weighing inside these 3 clusters on the proximal hand side, and very little, if any, weighing on the distal hand side. So we have the highest degree of asymmetry inside the clusters and no asymmetry between the clusters. So this is the end stage, but the question is, how do the proteins end up there? And to get there, we have multiple possibilities of inhibition, either across the cells where you inhibit yourself, or inside the cells where you inhibit the green complex, inhibit the purple complex from accumulating. And there could also be a serious set of positive feedbacks where you either accumulate yourself inside one cell or accumulate the other complex across the junction. So the question is, what's the role of cluster formation play in PCP signaling, and which of the many possibilities of feedbacks are required to polarize clusters, cells, and the tissue? So to answer this, we kind of envisioned 3 scenarios for cluster formation that may give us some information about what's going on. One scenario, and you cannot really tell that from the example here on the right-hand side, is that in the end state, all the clusters, imagine these are the clusters that go down the junction between 2 cells, all the clusters, they end up in the same size. So if you now plot the distribution of cluster sizes, you will see one peak at this specific cluster size that's then being read downstream and emitting the signal downstream. Another scenario is that these clusters, they may move along the membranes, fuse with each other, and aggregate into like eventually one giant cluster. And if that's the case, we will have a coarsening mechanism going on, where if we look at any time point, we'll see a bimodal distribution of cluster size distribution with a lack of intermediate sized ones, some large clusters that have merged with other ones, and some smaller clusters that didn't merge, possibly because they were too far away from the bigger ones. An alternative scenario is that not only fusion is going on between these clusters, but also fission. A cluster may, by random, split into 2. And if that's the case, then we will have a ton of small clusters due to the splitting events. And we will then find a power law distribution of cluster sizes, with by far the most smallest one. So what we kind of envisioned here was that by looking at figuring out the sizes of these puncta along the junctions, we could gain information about the underlying mechanism that drives PCP polarization. So the way that I did this was that I looked in a living tissue, so in a fruit fly wing, roughly like 24 hours after preparing information, remove the pupal case, and then I find the wing, the developing wing, inside a wing sac, where you here see the transparent wing sac, and here you see like folds in the wing sac, and then underneath it you find the actual wing cells, and this is one of the PCP proteins endogenously attached to GFP. So you can now see how this protein accumulates along the junctions into clusters. And in particular, and you may also, it's a little hard to see, but you may also kind of weakly see that it kind of accumulates mostly down this way here and a little less down the junction on the other direction, and thereby we have the polarization. So distal is down here on the lower right, and proximal, the hinge, is top left. If we now zoom in, we can kind of enjoy what's going on even further. And we find that Wang accumulates into clusters of all kind of sizes. We find giant clusters, we find small clusters. This is for sure not one fixed size at this particular time point. I bleach it to kind of gain a little bit more detail about the sizes of these clusters. I studied the bleaching trace for all of the clusters in the images. Here I take one particular cluster, look at the bleaching trace on a turf scope, and I can then identify fluorophore bleaching steps as the fluorophore is turning off and on. And I can then count the number of Wang monomers in this particular clusters when I started the experiment. So for this case, I could take the initial intensity, divide it with the fluorophore step size, and I will then know that estimate that this cluster consists of 8 Wang monomers. I do this together with mosaic experiments in fly wings, where I tag Wang in GFP in one subset of cells, and then in the neighboring subset of cells, I don't have the tag on. So there are cells up here too, but they're just not tagged with GFP. And this is crucial because this is the technique that allows me to study the proximal and differentiate proximal and distal junctions. So I know that all of the fluorophore intensities along this junction is proximal coming from this cell, and all the intensities down here are distal from these two cells here. All right, and here you can really see how polarized this specific cell is, and also over here you can see polarity, and you may even see large Wang clusters on the proximal side and small clusters on the distal side. Okay, so what I find when I look across like 17 wings, look at protein cluster sizes for flamingo, so each line here is one wing, and then I plot basically a histogram of protein cluster sizes, number of molecules in clusters, so the number of flamingo molecules on the proximal hand side in clusters. So what I see is that the most clusters are small with 1, 2, 3 monomers, and it forms an exponential distribution of cluster sizes with on average 4 flamingo monomers inside an average cluster. And on so the exponent up here gives you an estimate of the mean number of monomers inside the cluster. I do the same on the other hand side, and as expected, I find that flamingo is symmetrical on the two sides. Okay, so, but this surprises a little bit because an exponential distribution is none of the 3 cluster size distribution that we kind of expected when starting the project. So how do we get an exponential cluster size distribution? That can be explained by the on and the off rates being independent of the cluster size. So no matter how many there are, then the rate at which new ones comes in, or the rate at which the current ones they leave, it doesn't depend on how many are already there. That's one way of getting exponential cluster size distribution. So moving on to polarized clusters, I now look at Frizzle and Prickle that are on the opposite sides to each other, And here you'll see that there's almost no fissile or peggle on one side, whereas almost all of it is on the other side. So they basically almost act as a mirror compared to each other. And fissile and peggle are almost perfectly polarized. We do occasionally see like monomers of peggle on the, so to say, incorrect side, or monomers of fissile on the wrong side, where it's not mostly, but most of it is highly polarized into large clusters. And fissile is almost on the same order as flamingo. Vang, though, it exists in even higher numbers than any of the other proteins. And not only that, but we also do find Vang to some numbers of Vang on the distal side, something that has not been noticed in earlier confocal experiments. Okay, so the next question that we digged into is, okay, so we now find clusters of all kinds of sizes, but does cluster size matter? How does it matter for downstream reading this? And here we took inspiration from a paper that came out a few years ago, where they introduced a set of point mutation in the DIX domain of disheveled, one of the membrane proteins that accumulates mainly on the distal sides. And they found in vitro that this domain mediates clustering. So in the wild type, they see a full activity of beta-catening. When they remove the domain, there's almost no activity. And then when they introduce a series of point mutation, they find a gradient of activity of beta-catenin. So we developed this set of flies and did a series of similar experiments in vivo, where in our control flies, we see normal hair polarity, and we see relatively large clusters of Vang in the flies, whereas in a series of point mutations here in the DIX domain, we start to see a weak phenotypes in the hair polarity, slight reduction in cluster sizes, and other phenotypes have even more severe phenotypes and even further reduction of cluster sizes compared to our control. So this tells us that decreasing cluster sizes corresponds to decreased polarity. So it's essential to make large clusters in order for a correct downstream reading of in PCP signaling. So what I haven't told you at this point is how do PCP clusters then become asymmetric? Well, everything I told you this far just kind of tells you something about what's the distribution of cluster sizes. But now I return to the series of possibilities of feedback mechanisms that may be going on and kind of break the symmetry of a cluster dynamically during cluster growth and cluster decay. And to address this, I reduce the network here and move into mathematical modeling, where we reduce basically the green proteins here down to like one component here called F, and the purple proteins here down to one component called V. And there's in this network now, I try to model the dynamics by two feedbacks, intracellular inhibition between green and purple, and intercellular promotion between green and purple. And so now we have like a green complex that can enter and leave the cluster, and a purple subcomplex that can, a protein that can enter and leave the cluster with a certain rates. And then we know from earlier studies that fissile and disheveled, they have a bias influx to one side. So here I have a different rate on the proximal side for the green complex. I can write up a set of differential equations, and I can then run this model here with these four parameters: the influx rates that's independent of cluster size, the bias in influx for one of the proteins, and then the two intercellular coupling constant and the intracellular inhibition. You should now envision that this here is one cluster that with the two proteins. So here on this side, you have cell 1, the distal subcomplex. You can see how it enters and leaves, the green and purple enters and leaves the subcomplex. And then on the other hand side, you have similarly the two subcomplexes that enter and leave stochastically based on this set of differential equations and a specific set of 4 parameters choices for the 4 variables in the equations. And what we find here with this model is that the cluster orientation, it can be incorrect, turned the incorrect way for, in particular, small clusters, whereas large clusters, they tend to be oriented correctly. And now we should imagine when I do the experiments, I look at a random time point, say 5,000 here, and now I'm looking at not only one cluster, which is shown up here in the graphics, but I look at thousands of clusters, and I can now plot the distribution of V on one side, and I can plot the distribution of Wang on the other side, and I will then find that in the mathematical model, I will find that these tend to polarize with this set of parameters for this set of equations. So a simple possible growth law is sufficient to explain the data I've shown to you. I now turn into 2-color experiments where the mathematical model would predict that when we look across the junction to look at the 2 sides together, I will find that the combined sum of V on the 2 sides correlates with the combined sum of F on the 2 sides. And I can do this experimentally. And when I do this and tag one protein Fristel in M scarlet and match the green and the red to each other, I do find with some noise that Wang correlates with Fristel when I look at the 2 sides together. I can also mathematically make prediction on how one side of Wang would behave compared to the other side of Wang. So what's the number of proteins of proximal Wang for any particular cluster, and this at the same time, how much distal Wang is in that same cluster. And when I do this mathematically from one particular choice of parameters, I will see that they anti-correlate with each other. And I can do this experimentally also, and do these twin clone experiments where I have mosaics of green and red next to each other. So here I tag 2 purple proteins, Prickle in one subset of cells and Wang in another subset of cells tagged in M scarlet. And I do find experimentally that the mathematical prediction is confirmed that distal Prickle anti-correlates with a proximal Wang. Here I show the number of wings and the number of clusters looked at, and then the correlation coefficient between the 2 proteins on the 2 sides. So with this weak intercellular inhibition and intercellular coupling is sufficient for polarization. So with this, I have now studied a wild type scenario where the larger the in rate is, this is kind of related to the DIX experiments I showed earlier on, I see large polarity, whereas we should imagine when we go down and the influx is a little inhibited in the disheveled oligomerization experiment at DIX mutations, I could envision that the cluster size is smaller, the in rate is inhibited, and then also the polarity is smaller. So kind of you need large clusters to make to get a large polarity between the 2 sides. When I remove one protein in the mathematical model, I find no asymmetry independent of what the in rate is or what the outflow is. And there's also another experiment I didn't have time to talk about where we kind of plot the connection between the 2 subcomplexes. And in that scenario, we do experimentally see even a weak polarization, and the mathematical model also predicts that for large influx weighted. So what's more comes out from this model is that not only does the degree of asymmetry increase with the cluster sizes, but also the orientation of cluster sizes tends to be more correctly for larger clusters. So here the blue is wild type. So the fraction of incorrectly oriented clusters goes down the bigger cluster we look at. Whereas in the null loss of function experiment, it's like 50-50. We don't know what way they are oriented. And then when we cut the coupling between the proximal and distal subcomplexes, we still see a weak polarity, and also some clusters are oriented the correct way compared to the complete loss of function, but not all of the clusters as what we will see in wild type. So in wild type, larger clusters are more asymmetric and more likely to be correctly oriented. So with this, I will conclude that the on-off weights are independent of cluster size. Decreasing cluster size decreases the polarity. And large clusters tend to become more correctly oriented compared to smaller clusters where it's more 50-50 what way they are oriented. And then this mechanism that I told you about today would then become tolerant to errors and noise. And with this, I just want to thank all the people involved in this project, in particular the Alex Dunn lab at Chemical Engineering at Stanford, and Jeff Axworth lab at the Department of Pathology at the School of Medicine, also at Stanford, and our funding sources. And with this, thank you for listening, and I'm open to questions.
Great. Thank you so much, Silas. That was a great presentation. And while people are typing in their questions, I'll start off the Q&A with one of my own, which is in one of the early images you showed of the purple and green, and it looked like the clusters were evenly spaced, or that seemed like something that was consistent. And then in later ones, they're not evenly spaced. I was wondering if the distance between clusters is meaningful.
Yeah, that's a good question. So we have looked a little bit about so most of all the experiments I showed you today were kind of bleaching experiments where I bleach on the order of a couple of minutes. I've also done longer imaging, live imaging on the order of a couple of hours. And what I generally see is that these clusters, they don't move much amount. They're very little movement. They're pretty much like where they are formed, they stay there. I don't see, it's not fusion and fission, it's not dominant processes in this process. And then talking about the spacing between clusters, I would lean towards that they are close to be like evenly spaced, but like what dynamics, I don't know. We don't know like what dictates that here is going to be formed a cluster, and what's like dictates that at this and that distance is not going to be formed a cluster. We don't have any information about that.
Cool. All right, I have a question from Vidal in the audience. Do the DSH-DIX mutations also affect the clustering of PK-DSH result in your model, or only Vangel?
Oh yeah, so yeah, we looked at I quantified not only Vang, which is what I'm showing here. The reason I'm showing you Vang is because the earlier experiments from up here where we find there's the most Vang in these clusters. So, but I see the biggest effect when I look at the cluster sizes in the DIX mutants for Vang. That's where I see the biggest effect in cluster size decrease. But Frizzle does also decrease, not to the same degree, but also remember that there is less Frizzle compared to Vang. Frizzle decreases. I've looked also a little bit at Flamingo. It seems like all of the other core proteins also decreases to the extent that I have imaged them. It's not only Vang.
We have a question from Ashley. If it's essential to make large clusters, does this need to happen within a certain time window? And is there a point of no return where polarity will still be messed up?
Yeah, so I didn't have much time to talk about time today. So I imaged these clusters, and I said like on the order of like 24 hours after preparing information. I have looked at not only this specific time window, I have also looked like a handful of hours before and a handful of hours later. And what I see is that the cluster sizes increases slightly with the age of the wing. There's a slight increase in cluster sizes. We don't know like the underlying mechanism that make them increase, but we do see that clusters tend to become bigger with time, and they have the biggest size on the order of like 28 hours after preparing information. And this is also the time point where the pre-hair starts to form. So this kind of matches the hypothesis that you need large clusters to polarize the tissue, because the largest clusters are on the order of 28 hours after preparing information. This is where the hair starts to come out. And then the clusters stay around for a few more hours, and then they disappear. In loss of function experiments, I have done similar kind of removing a complete proteins and looking at the cluster sizes. And there I don't see this. I see they still form clusters, but they don't become as big. They don't increase with age. I don't know whether this answered the question.
I'll have Ashley follow up in the chat if there's more to ask. We have another question here from Earl Godfrey. How do you get differential expression of the purple and green clusters in nearby cells, electroporation or injection of constructs into cells?
So are you asking about the set of experience down here? If that's the case, then this is Drosophila genetics, where we in a subset of cells genetically can encode. This is like a toolkit in Drosophila specifically, where we can genetically encode that we have the fluorophore in one subset. So we cannot control exactly what cells that gets the fluorophore, but we can control that we get mosaics of cells, and then we are kind of just scanning the entire wings, and then we are looking for boundaries where these two fluorophores, they are fully on in one set in one color in one set of cells, and in the neighboring set of cells they are fully on in the other color. But it's by random which cells that gets what color.
All right, I actually have one more question, I think, to finish out the session, which is that how many clusters is sufficient? Like if you had one really big cluster, is that enough?
Yeah, that's a great question. And I don't know the answer to that. So these experiments are kind of telling us that the majority of clusters are tiny. And what's needed are the large clusters, and those are few. But whether you could just kind of place, like artificially place one cluster at one junction, and that just only, and whether that would kind of polarize the tissue correctly, I don't know for sure. Yeah. But yeah, it's like a few clusters that this is kind of hinting towards, that a few clusters are kind of the being read downstream, and how they are being read downstream also we don't know that exactly, what mechanism that kind of differentiates large clusters from small clusters.
Cool. All right, actually there's one more, and this is gonna be the last question. It's from Vidal. Are there any molecular models of PCP that account for the observed stoichiometry of molecules at clusters?
So you're asking, so the stoichiometry here, I'm not sure if I get the question correctly, but the way I kind of get the question is, so the stoichiometry here is, we had like long discussions about whether the stoichiometry, so here it looks like a 101 stoichiometry between flamingo on the two sides, like maybe roughly 101 between a flamingo and Frizzle. And then what is this? This is like 6 times as much weighing on one side compared to flamingo. Like whether this stoichiometry is probably not is tightly fixed. There's some variability in the stoichiometry. It's also not like, I don't think it's not like fully loose. Like wild type, I don't think you will find like a ton of flamingo on one side and then non-Wang. There's some flexibility in the stoichiometry. And this is probably allowing some plasticity in correcting for mistakes during cluster growth and during cluster decay. And so the stoichiometry and the plasticity in the stoichiometry might be important here. That's also what the mathematical modeling here hints towards. When gamma is too weak, it's too loose, the connection between the two sides, and you don't polarize as well. Yeah. Time is running, I see.
Yes, yeah. So if there's any more questions, please feel free to contact Silas. I'm sure he'd be happy to answer them. And with that, I'd like to thank Ashley and Silas for their excellent talks. The seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, January 10th, which will be featuring Suresh Damodaran from Duke University and Natasha Shiloh from the Stowers Institute for Medical Research. Thank you all for coming.